The purpose of this videotape will be to reinforce some of the various information that you have gained in reading the material in aspects of muscle physiology and how this muscle physiology relates to cyclic mandibular movement. You're very familiar with the histology of the muscle fiber and its relationship neurologically at the neuromuscular junction, and we know that this junction is what is electrically necessary in order to provide for a muscle activity. We know that the neurological input to the muscle is an efferent, or movement away from the central nervous system. We also know that there is such a thing as a reflex loop, and the purpose of the reflex loop is then to inform the brain as to what has gone on in the periphery. This reflex loop is very evident within the muscle system. In other words, there has to be a sensory arm of muscle activity in order to provide the brain information that the message has been received by the periphery and the particular action that was required by the brain has been taken care of. The sensory ending that provides this information to the brain is the neuromuscular spindle, or as it's abbreviated, the muscle spindle. And if we look at the first slide, we'll be able to see some of the anatomical configurations of this very refined and sensitive sensory ending. First slide. The muscle spindle is a bag of connective tissue, and within this connective tissue sheath we find two sensory fibers, a long fiber that is swelled in the equatorial range, which is called the nuclear bag fiber, and a smaller fiber adjacent to it that is thin and narrow in the equatorial range, and this is called the nuclear chain fiber. Within a muscle spindle, there may be as many as 20 different fibers composing nuclear bag and nuclear chain. The muscle spindle is found in parallel with the muscle fiber. We can refer to the muscle fiber itself as the extrafusal fiber, and we will refer to the nuclear chain and nuclear bag fiber as the intrafusal fiber. Next to the eye and the ear, this muscle spindle, or nerve receptor, is the most sensitive nerve receptor in the body. It has both a sensory as well as a motor input to it. This is one of the few receptors in the human body that has a dual sensory and dual motor innervation. The sensory arm of the receptor involves two fibers, a rapidly conducting large fiber and a slowly conducting secondary fiber. The rapidly conducting fiber, being the more sensitive of the two, is found within the equatorial range, and this is called the primary ending. The secondary endings are found within the periphery of the intrafusal fiber, and the sensitivity of which these fibers respond is that of stretch. When a muscle is stretched, it tears its insertion away from the origin, and the protective mechanism is for the brain to cause contraction within that muscle so that the origin will not be pulled from the insertion. We call this activity a stretch reflex. In other words, if we were to tap this tendon, it would cause an elongation of this muscle, which would cause an elongation of the intrafusal fiber as well. This would fire the primary ending, whose cell body is located within the dorsal root ganglion. principal fiber then would then progress to the central nervous system, the brainstem or spinal cord, and it would make connections either on an interneuron or an alpha motor neuron. The alpha motor neuron then would then cause activity to occur at the neuromuscular junction, and contraction would take place within the muscle, causing a reflexive protective activity whereby the muscle prevented this stretch or elongation. We refer to this as the myotactic reflex, and it is a one synapse reflex. It is a monosynaptic reflex. You must also realize that when you do a muscle stretch activity, such as tapping the patella tendon, not only do you have to turn on the agonist, you also have to turn off the antagonist. So the process with which the monosynaptic activity takes place is very highly simplified in this diagram, because it does involve the inhibition of the muscle that would serve as the antagonistic type muscle. We said that this neuromuscular spindle had a dual sensory and dual motor innervation to it. If it has a motor innervation, then this tells us that these intrafusal fibers are capable of muscle contraction, and that is the case. They have an actin and myosin-like compound within them that is very similar to the actin and myosin activity of the extrafusal fibers. Not only do we have a protective mechanism whereby the muscle is protected with the neuromuscular spindle, but we also have a way of monitoring via the central nervous system, the brain, and other brainstem centers, of controlling the degree of stretch and irritability. In other words, we do have a way of setting the thermostat for muscle activity, and this is provided through the motor mechanism of the intrafusal fiber. This is the way it works. The cortex or the brainstem can send down activity from higher centers to the area in the ventral horn that houses the gamma motor neurons. The gamma motor neuron is a smaller neuron than the alpha motor neuron. The gamma motor neuron specifically goes to the intrafusal fiber. The firing of the intrafusal fiber will cause the entire muscle spindle to contract, making it much more sensitive to muscle elongation. Once the contraction in the intrafusal fiber takes place, then the thermostat of muscle activity is set into place. Then only the slightest amount of change in length of this muscle will cause a firing of this muscle spindle, and it will then progress through the dorsal root ganglion and into the ventral horn to the alpha motor neuron, and again causing reflex muscle activity as a protective mechanism to protect the muscle from elongation. There are also sensory receptors found in the tendons of the muscle. This is called the Golgi tendon organ. The Golgi tendon organ not only informs the brain of stretch, but also contraction. So within this muscle, the sensory aspect is the degree of stretch is programmed through the muscle spindle. Stretch and contraction is programmed through the Golgi tendon organ. Of the two receptors, the muscle spindle and the Golgi tendon organ, the neuromuscular spindle is by far the most sensitive. Not only does it give the degree of stretch, but it also will inform the brain as to rate of change of stretch, this being very specific. If we were to look at a cutaway of what is going to happen we would see that the neuromuscular spindle has its cell body within the dorsal root ganglion. It sends out a proximal process then that will pass through the substantia gelatinosa and into the ventral horn to end on a ventral horn cell or an alpha motor neuron. The higher centers then would program into the area where the gamma motor neurons are located. This would involve a typical spinal nerve. Muscle activity to the knee, to the elbow, to any joint would have a proprioceptive or joint sense or muscle sense aspect to it. But this also involves muscle activity in and about the head and neck, and specifically within the temporomandibular joint. The context of our next videotape will be to discuss applications of muscle coordination with cyclic mandibular movement. The purpose of this videotape is to familiarize you with the aspects of muscle physiology as it relates to cyclic mandibular movement. In part 1, we discussed the typical spinal nerve. We also discussed the monosynaptic or myotactic reflex. Now we are going to apply this information to the muscles of mastication and the movement in and about the temporomandibular joint. You know that the trigeminal nerve is the principal sensory nerve of the head. It has a ganglion, the Gasserian or semilunar ganglion, that is located within the middle cranial fossa. The trigeminal nerve has 3 branches that provide sensory information as a reflex arm back to the brain from the periphery. The muscles of mastication are also innervated by the motor arm of the trigeminal system. The way in which the motor arm receives its sensory input is somewhat different than other spinal nerves. The area of the brainstem which receives information from the trigeminal system is called the pons. We have on one side, in blue, the sensory portion of the trigeminal system, which consists of the Gasserian ganglion, the principal sensory nucleus of the trigeminal, and the spinal nucleus of the trigeminal. On the opposite side, we see that there is a motor nucleus of the trigeminal nerve, and it is within this motor nucleus that the alpha and gamma motor neurons are located for all of the muscle activity within the muscles of mastication. The portion of the trigeminal nerve which then transports the motor activity is the third division, or what we call the mandibular division. The proprioceptive or joint muscle sense within the trigeminal system, the muscles of mastication and the temporomandibular joint, do not have their cell bodies within the Gasserian ganglion, and this is one of the unique features of this cranial nerve. On the other hand, the cell bodies which should be located within the Gasserian ganglion are located within the mesencephalic nucleus. This mesencephalic nucleus has true unipolar neurons located within it. These cells actually should be located in the Gasserian ganglion as they are in the dorsal root ganglion of a typical spinal nerve. The reason that they are not located there is that in the embryology and development of the central nervous system, these cells started their migration a little later than did the other sensory cells, and the process of development stopped before they could complete their migration to the Gasserian ganglion. So the cell bodies of the proprioceptive or muscle spindles receptors are not located here, but they're located within the mesencephalic nucleus of the trigeminal system. If we were to give a brief diagrammatic